So this week I sit down with Kaira Wagoner, a researcher out of the University of North Carolina, and what she has been working on is a project where they are detecting that bees are using a response that they're calling a unhealthy brood odor response. And I discovered, I was just sitting down late at night and just kind of came across her paper as I was doing some research, and she accepted my invitation to come on, and I'm super, super excited about this interview, and I think that you guys will find it incredibly insightful. And so I guess maybe let's just jump right into it. We kind of kicked this one off a little unorthodox, so enjoy.
So this is a video of hygienic behavior that I like to show. And if you look at the center of the video, you'll see these bees are pulling out an unhealthy brood from the colony.
So that's sacrifice.
Yeah, there's a couple in there. And that sacrifice, it's a small sacrifice that just really makes a big impact on the health of the colony overall.
So was this one of the colonies that you guys were studying, or is this just throughout your beehive inspections that you got this?
Yeah, this is one of the colonies. I pulled it out after doing a UBO assay. And so this was a different area of the colony where I just saw them pulling out brood. So I pulled out the camera and got a quick video.
Very cool. Now, something I guess I didn't ask is, where did you guys source the queens?
So a lot of my queens have been donations from Minnesota hygienic breeding programs based on freeze-kill brood, the USDA pole line and VSH lines. And then I get Italians from all over Georgia, South Carolina, North Carolina to compare.
Very cool. So it looks like was this within that test area, or, I mean, it looks like there's actually quite a few cells tore open in there.
Yeah, I don't think this was in the test area, but I don't remember. It could have been a freeze-kill brood. This is actually a video from probably 8 years ago, 6 years ago.
So, the reason I ended up getting in touch with you was because of that, the study that you've done on the Assay with that UBO. Do you want to explain just kind of in general what the study was about and then how it was performed?
Sure. So we essentially had found these compounds that are elevated in unhealthy brood. So it didn't really matter what the trigger was. So we found these same compounds were elevated in varroa infested brood, even if the virus level wasn't high. And they were also elevated in virus infected brood, even if there was no varroa present. But these same compounds were elevated. They're present in all brood. Every bee in the colony has these compounds present. But if they're unhealthy, they seem to be elevated. And so we turn that into a tool. We can actually synthesize these compounds in the lab, make them identical to the way they are produced naturally by honeybee brood. And then we can spray it onto wax caps and trigger hygienic removal with just a 2-hour assay. So we basically apply it into a small circle. We kind of demark an area using a small piece of PVC pipe, spray these compounds onto an area of capped brood cells, and then put the frame back into the colony for 2 hours and then pull it back out. And in a hygienic colony, you'll see sometimes 100% of those cells uncapped. They don't actually tend to remove the brood because once they uncap the cell, there's no more signal there. There's no more problem. And so they'll cap them back later. But what we found is we can correlate the reaction of the adult bees to that test, that pheromone test, with their resistance to varroa. So if we do a 2-hour test in June and a colony scores 60% or above on that test, so they uncap 60% or more of those cells in 2 hours, they tend to have much lower varroa loads in August than the ones that scored low on the test. And the difference is pretty substantial. So we had an average of 7.2% infestation in the low scoring colonies and below 2% in the high scoring colonies.
That was 7.2, you said?
Yes, 7.2.
Okay, I was trying to pull up a picture of that. And that's, the way that, there you go. And the way that you guys did that, is it identical in nature in regards to the PVC pipe as like the freeze method? And maybe I should explain the freeze method, or unless you'd like to explain the freeze method that has kind of been used to indicate or a way to indicate VSH qualities.
Sure. So the freeze kill brood assay, essentially what you do is you pour liquid nitrogen, which is very cold, onto an area of capped brood, and it freeze kills the brood underneath. And then typically the way the assay is done, you put the frame back into the colony for 24 hours, and you look at how many of those dead brood have been removed completely over a 24-hour period. And so we actually ran this study. We ran freeze kill brood and this UBO assay that uses the pheromones I talked about in the same colonies. And then we just looked at how well each of the different assays predicted the varroa level. And so what we found is there is a difference in both cases. So like I just mentioned, in the low UBO colonies that scored less than 60% on our UBO assay, these are all untreated for varroa. We saw an average of 7.2% varroa infestation. It was 1.6% on the high UBO colonies.
So just to be clear, the low UBO indicates that there's a low reaction to
exactly
something going on. And in this case, what is it, unpleasant brood odor?
Unhealthy,
unpleasant. Maybe to the bees it's unpleasant.
Also to the beekeeper. Yes, it's always unpleasant to have unhealthy bees. I like it.
Right.
Yeah. And so we saw also a difference with the freeze kill brood assay. We saw the ones that responded. The threshold for the freeze kill brood assay is 95% instead of 60% like our UBO assay. But if a colony removed 95% or more of that dead brood within 24 hours, they had a lower mite load. But what we found was the mite load for those high freeze kill brood colonies was right around that threshold that we call it sometimes the economic treatment threshold of where you're meant to treat for varroa.
So like the 3 to 4 mites per 100-ish.
Exactly. So the high freeze kill brood colonies had an average of 3.2% mite infestation on adults. So they're right above that threshold of where you usually hear it's time to treat. And that was for the high scoring ones. The low scoring ones were 6.8%. And so, what this told us essentially is the UBO assay is just a little bit better, a little bit more precise at distinguishing those colonies that are likely to survive on their own, that can handle varroa on their own, and those that really are going to need treatment.
Very cool. So just for the people that are listening, because some of, we'll publish this on YouTube as well as just through your typical podcast platforms. But to do the test, it's, about a 2-inch PVC, or do you have a specific size that you guys use?
Yeah, it's about 4 centimeters across the diameter. I can show you what it looks like for the YouTube folks. So here I have image of, on the left, what happens in a hygienic colony, and on the right, what happens in a non-hygienic colony. It's a little piece of PVC pipe. We actually use a lathe to cut away a small little edge, so you can press the PVC pipe in and gently turn it, and it basically just makes a small ring in the wax. And we spray the compounds on at time 0, put the frame back into the colony, and 2 hours later we pull it out. And you see something like this on the left in a hygienic colony. I mean, this was, let's see.
Which is almost every cell looks chewed open.
That's right. I think there are 2 in here that are still capped. So this received a 96%.
And I'd even argue that one is on the edge.
Yeah, so what we generally say is if it's 50% or more inside the circle, we count it.
Okay.
And so then on the right, in a non-hygienic colony, you may see absolutely no change, no response. You may see a couple uncapped, but there's a really clear difference between a hygienic and a non-hygienic colony. And this is a great way to be able to tell the difference between those 2 phenotypes, those 2 behaviors. And it plays out with your varroa loads.
Interesting. So did you, and this may be kind of a bit of an irrelevant question, but I'll ask it anyways. Did you see or follow whether or not the age development of the pupae made any difference whatsoever?
It doesn't. So we've looked at anything capped from, just capped to basically purple eye is fine. The one thing I do see, if you have brood that's about to emerge, for some reason, spraying this compound on seems to make them want to emerge early. So that kind of messes up your count. But any brood that's white, white cuticle, any age, there's no effect.
Very cool. Okay. And, we talked a little bit about this before we started recording. But one of the things, when I read this, I got very excited because one, it makes the possibility of testing for VSH much more readily accessible and done much quicker and easier, where I think the methods that bee breeders and scientists have been using now is you open a cell, you pull the pupa out, you got to go through 100 of them, you do mite counts, you got to be able to identify the difference between a reproductive mite, a unreproductive mite, so on and so forth. And so you had mentioned potentially that there's a conduit for this to become available at some point for beekeepers to use. Can you speak to that, or is it too soon?
No, absolutely. Yeah, we're working real hard on it. It's not available yet. I totally agree. So what we tend to see with the selection methods for hygiene in general, I think, is a trade-off between the feasibility of performing the assay, the efficiency of performing the assay, and the efficacy of the selection method. So the methods that are easier to perform tend to work, but not quite as well as the methods that are really labor intensive. And so what we saw was the need for a easy, rapid, and really precise method of measuring hygienic behavior. And so that's what we've come up with. We're currently working on developing it. So the system that we have right now is great for research, but we're, using these gas tight syringes and measuring out small quantities in the field. And so we don't have a shelf ready product yet. But we recently launched a startup that is working towards commercialization. So we got an NSF STTR grant that really is trying to help us de-risk this innovation, figure out if it's really the solution people are looking for, which I think now we're pretty sure it is.
I would say it is.
I'm glad to hear that. Yeah, we've had a whole lot of interest. I actually have, I think about 10, 11 collaborations going on right now of field tests in about 6 different countries. So it's not just in the States. People are really interested in using this as a tool for selective breeding.
I would be happy to test it. I mean, honestly.
No, noted. Yeah, we're excited about the level of encouragement we've gotten from the beekeeping community. And so we're working really hard to get something that's suitable for the shelf. Basically, we just our main, our mixture is ready to go, our pheromone mixture, but we are working on putting together an applicator that has a good shelf life and that makes things easy for running the test, and also hoping to make it easier for folks by putting together an app that can help you kind of score the test more easily and track the results.
Something like you take an image and it could potentially count how many are already open for you.
Exactly. Yeah. Even, I think a later generation might have some AI counting for you. But even a prototype that says, okay, take an image now of this, take an image now of this, and then you can tap on the cells that you want to count, and it just throws out a score at you and maybe
Very
gives
cool.
you some indications of what you should do based on that information.
So we kind of I directed us right almost to the finish line, but how did you discover the compounds? Well, first, what inspired you to go down this road? And then second, how did you detect ultimately what those, maybe I'm going to use the wrong word, pheromones are? And then how did you end up synthesizing them?
Sure. So would you believe it was mosquitoes that got me here?
Never, ever.
So I did a master's. I was studying some biomarkers of estivation, which is kind of like summer hibernation of mosquitoes. And so I got really into looking at these chemicals on the cuticles of insects. I decided to switch to honeybees for a PhD. And, at the time, a lot of people were interested about hygiene. They knew about hygienic nurse bees and the adult bees and what the adult bees were doing. But we didn't know a lot about, what it was they were detecting inside of the cell. So, obviously there's some kind of trigger in there that's inducing this uncapping and or removal behavior. But what was that? And so we started by infesting brood with mites. And then we looked at the difference in the chemical profiles of the...
You would place a mite in a cell?
Oh, yes. Lots of them. With so what we did is we took a little tiny paintbrush and we would introduce mites into cells. So cells that had just been capped within the last 16 hours. And we lift the cell cap up, we put a mite in, we close it back, and then we have a clear, those old school transparencies. We put a clear transparency on top.
For like a projector?
Exactly. Yeah.
Oh, interesting.
We mark where that cell is.
Anyone younger than us has no idea what we're talking about right now.
The projector. Yeah. No, it's actually a really useful tool, and then you just thumbtack it in, and you know exactly where that mite is, and then we would monitor removal over the course of several days, 8 days, and we saw that those with the mites were being removed. That's what we would expect, and then I started collecting them and just looking at the chemical differences. We use a compound called hexane. Like dissolves like in the chemistry world. So hexane is a hydrocarbon that just pulls off these hydrocarbons from the cuticle of the insect. It's something that all insects have. And we analyzed the ones that came from mite-infested brood and the ones that came from control brood that had no mite infestation. And we saw these tiny differences. It's not a novel compound. It's there on every single bee in your colony. It's just it's actually a set of compounds that are found in higher quantities when the brood is unhealthy. And it didn't matter, like I said before, if it was varroa infested or virus infected without a varroa, but those same compounds were elevated.
So, I mean, how do you detect after using this hexane what those compounds are? I mean, is there...
Sure. We use something called gas chromatography mass spectrometry, or GCMS. Wow. So it's a fancy machine in the chemistry department. Essentially, what it does, we inject a tiny amount of our extract into the machine, and it runs through a column. It heats it up. It separates the compounds, and it tells you which compounds are present and how much of each compound is present. And so it's a fascinating tool. And it took a long time looking for these differences, because at first we thought, we'll see something maybe different. We'll see a new peak, and there were no new peaks. But what we started to notice after running hundreds and hundreds of these samples is that there were just these tiny, tiny differences in the amounts. And the olfactory system, the ability of the bees to smell, is just so sensitive that they can detect these small changes.
That is fascinating.
One really cool thing I'll tell you is that in 2017, a paper came out and showed one of the same compounds we had found associated with hygiene and unhealthy brood in honeybees is also elevated in ants that are targeted by a similar behavior in ants. They call it unpacking, but the ants actually emit a similar compound when they're unhealthy, and it triggers something similar to hygiene in the ant colony.
So I don't know anything about ants. Do they raise their young similar to bees do in cells, or?
I'll be honest, I don't know a whole lot about ants either. I don't know that they, I think there are some species that raise them in cells and others that don't. But yes, in this case, they were unpacking them from a cocoon, similar to the cocoon that honeybees spin inside the cell.
Interesting. And then I would imagine once you've determined where those peaks were, or which compounds were elevated, I'd imagine it's no easy task to produce that chemical as well, or a compound.
It's not, and that's where my chemistry friends come in, because they like to play around at the lab bench all day. I like to be out in the field with my bees. So we actually collaborated with some great chemists, Jocelyn Miller out in California, and more recently here at UNC Greensboro in North Carolina, and these brilliant chemists can take a whole bunch of starting materials that look nothing like what we want and do some magic in the lab, and they come up with these synthesized chemicals that are exactly the same as those produced by the honeybee, but we no longer have to kill the brood and extract tiny quantities at a time. We can make it synthetically, which is much nicer for the honeybees.
So do you anticipate, what is the commercial product? Do you guys have, I guess, a vision for what the commercial product looks like and how it would be implemented? Or is that something you guys are still working on?
Yeah, absolutely. I think our mixture is ready to go. Like I said, it's just figuring out that applicator. I think optimistically we'll have something by next summer. It just depends on how long it takes us to get this, this whole entrepreneur thing going. We have to still source who's going to be making the compounds for us, and who's going to be doing the mixing, and what we're going to apply them with in terms of the applicator. But we're working really hard on it. We're dedicated now. Our startup company now has 1.5 full-time employees.
Congratulations.
Thank you. It's a big step. Yeah. So we're really excited, and we're actually building already a list of people who are interested in staying up to date with the project and who want these compounds. People are, begging us to come test their colonies and to be kind of the first ones to get the compounds when they're ready. So we're pushing as hard as we can, and hopefully we'll have something for folks by next year.
Right. Well, obviously, I'm sure you're aware, doing it right is more important than getting it out there quick.
Absolutely.
That's the boat that I am.
Could not agree more.
posting my flag in at the moment as well. I've been working on my project for what feels like way too long already.
Right. And that, I mean, part of that is the way we're addressing that is having all these collaborators through other research organizations, a couple commercial beekeepers, and some other universities that people are field testing this now. I've been doing this and saying it works for a while, but it's nice to kind of get some tests out there to other people so that they can run field trials and give us feedback. So we definitely want to do it right the first time.
Right. Well, I am insanely excited about it. And, one of the things that, again, we talked about before we start recording was I always felt that freeze method, at least in my mind, I mean, you're showing that there's some correlation between the freeze method and this UBO. But in my mind, I always was like, well, the freeze method, we're really detecting a dead bee, that hive is capable of ejecting a dead one. Well, in a scenario like varroa, that bee is still alive. And so at least, and I'm not a researcher, but logically I was like, this doesn't seem like, it seems like we're hitting the target, but we're not near the bullseye yet. And when I read your research paper, once again, was like, they hit it. They absolutely nailed it. And that's where I got super excited about it. And to be honest, I was like, how can I make this? How can I synthesize this? How can I sell this? Because if it becomes this easy, I'm a proponent that, we need to be treating less, if maybe not at all, for varroa and raising bees that have this behavior so they can solve the problem themselves. And then once we get enough not genetic diversity, but once the gene pool's flooded with genes that express that behavior, we start to solve our own problem. And I think I can't recite that, what took place in Africa, but Africa did it. They said, we're not treating our bees anymore. And 3 years later, they have no problems. And so,
well, I think it comes down to a sensitivity thing too, with the different assays. So, I think that, it's pretty obvious that at least over time, a dead bee is going to emit a stronger smell than one that has, a small parasite or even a large parasite on it. It's going to be more of a subtle cue when the brood is still alive. And so I think, you can select for the same behavior, but you can maybe select for bees that are even more sensitive to the smells that trigger that behavior,
right.
Using something that has these more subtle pheromones compared to, dead brood. We know from past studies that the dead brood emits oleic acid. So there are a whole set of compounds coming out of dead brood that are different than you might find in living brood. And so it's just a more subtle test if they can distinguish small increases in a compound compared to these new compounds, or at least
right.
compounds at a much higher level.
Which I would then argue that freeze method really isn't doing as much of anything, unless that brood, for whatever reason, is in such dire distress that it's able to emit an incredibly strong.
Well,
would it be a pheromone? I mean, are they pheromones?
Yeah. Some of them are pheromones, but I wouldn't say that it's not doing anything. So what we found in our study, there was still a pretty big difference between the high freeze kill brood and the low freeze kill brood. So the difference was 3.2% varroa infestation in the high freeze kill brood and 6.8 in the low. So, I mean, that's more than double. It is making a difference with varroa, but it might not make a difference with whether you have to treat or not. You may still need to treat to pull your bees through the winter.
Right.
So I think there is a difference, but maybe we can do better.
Correct. Well, and I'm obviously in the camp that treatment free is going to get us there faster, and so anything that kind of leans us a little bit more that direction, I'm for. Because I was the guy that wouldn't spray his lawn with anything forever, and now I find a need here and there. But relying on chemicals, like I think we just in general have. I mean, you look at even the monocultures that we've developed in agriculture, it's like that seems to be the number one go-to solution for our problems. And, often nature already has a cure. We're just too impatient to wait around and discover what that actually may be,
Right. And, what I'd like there to be is an easy way to do the better thing. And so far that hasn't been an option, but we're hoping this is it. And I won't say I don't think it's a silver bullet. I don't think we're going to just all of a sudden not be able to use or not need miticides. I think it's going to take time, but if you're selecting for hygiene, if your neighbors are selecting for hygiene, so that when you requeen, your daughter queens are requeening with hygienic drones, that's going to make a big difference. And we have shown that in a study that I can't show the results for yet. We're in the process of still collecting data and publishing, but I'll have it out there soon. And we do see that this trait is heritable, and that the mom is really important, the origin of the egg that you raise your daughters from, but that drones also play a big part. So, raise hygienic queens, but also convince your neighbors to do the same.
Right. Hopefully, I won't talk about neighbors. We already know that story. But yes, I totally couldn't agree more. And I'd love to have you back on once you finish that research, which sounds like you're pretty darn close to. So
we're getting there. Yeah, absolutely. I'm happy to come talk bees anytime.
Great. And one of I know like the effort is there. Well, maybe that's not the right way to put it, but if you look at the landscape and you've got just little dots of people that are performing or that are really emphasizing VSH qualities in queens, does each, if they're reintroducing new genetics that possess the VSH qualities, over time, have you guys seen any or read or done any research on how that may be positively influencing the surrounding apiaries?
Oh, I'm sure it has an effect. But I think at this point it gets diluted quicker than it has a positive effect, just because there aren't enough people selecting for hygiene. But I think we'll get there. I think also that the US is a little bit behind some other countries, Canada, a lot of Europe. They started selecting for hygiene, I think, before us, or at least more people have adopted it.
I follow a fair amount of it, and I've actually just released a podcast today where I've been trying to do like news release stuff like every 1 to 2 weeks. And Canada actually isn't, most of Canada is kind of not seeing great numbers. However, though Nova Scotia, they've got great survival rates. And then now what's going on in Australia now that they've got the varroa mite.
Yep.
And I sadly think that it's a bit of a lost cause, because once it's there, I just don't understand how they would get rid of it. And so it'll be interesting to see how they react to it.
Yeah, we're hoping to get a couple projects going there this year too. So they were interested. Australia has been wanting to select for varroa resistant bees even before varroa came, just so that they'd be ready, but they don't have a great way of testing for whether or not their stock is varroa resistant, because they don't have varroa,
right?
Or at least they didn't. So we're hoping to get a couple of projects going there and help them build up some immunity, some hygienic immunity before varroa really sets in. So
Well, that would be an interesting study, would be to get into an area, for instance, Australia now, where there aren't any mites, and do these tests with the UBO just to see like what those results look like there versus an area that has varroa, and if maybe there is a increase of VSH in the genetic pool in areas that have varroa versus areas that don't. Maybe there's no difference. That would be interesting to see, but we're gonna run out of that time, because eventually Australia is gonna have it everywhere.
Yeah, unfortunately, that's probably the case, but
Yeah,
well,
unfortunately.
hopefully they can hold it off a little bit longer.
Right. And right now it just sounds like it's in I don't even know part. It's the southeast corner of the country, which I think is New South Wales, I believe. But I'm not real great on geography.
I want to say Newcastle, I think is where Port of Newcastle, I think is where they found the varroa.
That sounds right, because I think they have a port there, and I believe they had several apiaries that were specifically set up just to detect varroa. Which
they've known it was probably coming, so hopefully their intervention will prevent the spread, but we'll see. Time will tell, right?
Yep. So in your city, was there anything that really stood out to you that kind of surprised you throughout the process?
Is there anything? Yeah, I mean, I think the first thing that surprised me was that these weren't novel compounds that appear when the brood is unhealthy, that they're just these compounds that are present on every bee, and these small changes are what the bees pick up on. It's just impressive, their level of sensitivity.
And so it's almost as if they're the VSH. Do you call it a VSH quality?
No, I try to avoid that term because I don't think it's specific to varroa. So some of them, I'll share some of the most recent data, also something we haven't published yet because we're collecting more this season. But we saw that the virus levels in the colonies that tested high UBO, so the colonies that were reacting to these pheromones and uncapping in our UBO assay, those colonies had lower virus loads than the colonies that didn't score high on the assay, so that are less hygienic. And what was really neat was some of the differences we saw were in viruses that aren't related to varroa. So of course, deformed wing virus has a strong link to varroa, the varroa mite, but there are also viruses that were lower in the high hygienic colonies that are completely unrelated to varroa. They're not vectored by varroa. And so I really do think it's more of a general response than a varroa specific response.
Now, do you think that those viruses or other illnesses, I'm assuming they emit some sort of smell?
Yeah, again, I don't think it's the virus itself or the varroa itself that emits the smell, but the stressed pupae or the stressed
I
brood.
should have phrased it that way. My apologies.
Yeah, so I think the same thing is going on. The brood is unhealthy, and that's again why we decided to call it the unhealthy brood odor assay, is because the brood is unhealthy, whether it's varroa or virus, and their response, altruistic or not, intentional or not, that is their physiological response, is to produce those compounds in a higher quantity, and the nurse bees can pick up on that and do something about it.
So what if maybe you guys have come up with a name? So what quality would you assign this to the bee, if not VSH?
I think that's a great question. I mean, I think it's sensitive hygienic, maybe just not the varroa sensitive. So I think they're just essentially a higher quality hygienic bee.
My mind's going crazy now. I'm thinking maybe like healthy brood sensitivity or something at HBS.
Yeah, send me all the ideas. I need all the
I'm going to roll with HBS for now.
There you go.
Healthy
I like it.
brood sensitivity.
I like
Or maybe unhealthy brood sensitivity. UBS.
it. UBS.
But isn't that like, that sounds like a bowel syndrome.
Whatever it is, it's useful.
Right. Well, and I hesitate saying VSH ever since I've read your study, because it's like, I don't even know. Like, it just seems.
And part of the problem is the VSH have been selected for different, using different methods, and the terminology can get really confusing. So
Well, and the reason I've been thinking about it is, I'm intending on putting a article or a blog post together that kind of starts to lay some of that out, because I think as researchers like you start discovering more detailed aspects of what we're after, we really need to start because even someone like me could start saying VSH when it's really not that, and then you're kind of pushing a bit of a false narrative unintentionally, and so it'd be nice to come up with a name for it, and you get to name it.
Yeah, let me know what your ideas are. I would love to hear them.
I'll think about it a little bit. I don't know that you want any of my ideas. So.
Sure, anybody's ideas are welcome.
Yeah. So if you're listening now, in the comments as we're talking, and you're screaming at your computer or your radio or your phone or whatever the heck you listen on now, just type it in the comments.
Yeah, I want to hear it. Absolutely.
All right. Well, great. Well, do you want to share anything else in regards to, well, I guess anything bee related?
Anything else? I have so much to talk about. Unfortunately, I really have to go. My nanny's here with the kids. I can talk bees for an hour. That's the problem.
Well, we will definitely schedule the next one. Look for an email from me, and maybe in, 2-3 weeks we get back on and kind of pick up where we left off.
Yeah, that'd be fantastic.
Do you think you'll be done with your other research by then?
Oh, I'm never done with research.
Well, no, the one with that you kind of glimpsed, or you showed me in regards to those 2 apiaries.
Yeah, I should be done collecting the data. We'll put together the manuscript this fall and submit it. I'll be doing a bit of traveling to Europe meet with some collaborators in Belgium and the Netherlands. And I'll be back in October and writing up these manuscripts. I always try to publish open source so that my work is available to anybody. I don't want you to have a university connection to be able to read the work. So I do work hard to publish everything open source. I pay more to be able to do that because I think it's important to get the word out there. So when it is published, you should all have access.
Wonderful. Well, do you want to direct anyone to website, any of your research?
We are working on a website. It's not up yet for .com, but we will hopefully launch it. If you do want to get on our mailing list, we haven't even sent out a first mailing yet, but we are compiling a mailing list. You can contact me at Optera, LLP, OPTERA LLP@gmail.com, and I can get you on that list. So the plan is just to send out occasional emails about project updates and where we are and what we found, and keep people in the loop. So that if anybody's interested, they can get access to the compounds as soon as they're available.
Great. And we'll include all those in the show notes or the description so people can get a hold of you.
Excellent. Well, I really appreciate it and look forward to the next time. It's great talking to you.
Yeah, you too. Take care.
All right. Thanks so much.
Yep. Bye.
Bye-bye.
There was nothing left to say. How to get up, walk away. Know when to hold them, know when to fold. Know when to hold them, know when to fold.